Okay. So hello, everyone. Thank you for joining today's discussion. I'd like to quickly introduce Thomas as well as our team. So, yes. So Thomas is a scientist who really know what he's talking about because he's been working with farmers and plants for over 30 years. While not working with farmers, he is a visiting professor at West Anhui University in China. And so he's been working with ComCat for over 3 decades already. So today he'll give us a brief lecture on what ComCat is, what does it do, its mechanism, and share some of his stories working with farmers as well. And most importantly, he'll be answer a lot of your questions that you might have. So please bring question and ask Thomas. We'll also have Dr. Jai and Jim joining us. So Li John, Dr. Jai is our HR manager. So if you have any HR related questions, you can ask her. We'll also have Jim. He's our warehouse and logistic manager. So anything that is logistic or related to the warehouse, you can ask Jim. Alrighty. So without further ado, Thomas, you want to get us started?
Oh, can we also ask our candidate, our partners to very briefly introduce them so Thomas know each of them what they do? Yeah, thank you.
All right, Will, you want to start first?
You want me to start?
My name is Will Pagel. I live in Edinburgh, Texas. Last 11 years, I've worked for Wilbur Ellis as a sales agronomist down here in the valley, dealing with citrus, row crop, veggies, melons. So it's been quite an array of products, a lot of specialty and high value crops. Before that, I was in Iowa as an agronomist, dealing with mainly corn and soybeans, a little bit of alfalfa for growers right there in central Iowa, the I-80 corridor there. For that, 5 years, did corn research for Syngenta, Golden Harvest, Iowa, Kentucky, Hawaii, Puerto Rico, so all around. So that's my background. So nice to be here.
Will, can I have a short question to you? You work with Wilbur Ellis. Do you know Dr. Dave Schultheis? He was Dr. Dave Schultheis. He was a key researcher for seed treatments by Wilbur Ellis. Do you remember him?
I know Paul Johnson is in charge of seed treatments now. I wonder, has he been? Yeah. So
no,
I don't know.
It's 10, 15 years ago.
Yeah, like I said, I started 11 years ago. So Paul was the one I met. So he must have been already moved on.
No, but I worked very close with him for 10 years on ComCat, and maybe you remember Manifest. This was a seed treatment patent or product from Wilbur Ellis, and this was we developed together.
Oh, okay. Yeah.
Mark, do you want to introduce yourself?
Yeah. Super easy. Mark Taylor, West Texas to Arizona, just a farmer.
I like it.
All right. Kevin, do you mind introducing yourself a little bit?
Yeah, my name is Kevin. I'm out in California, Central Valley, Southern Valley, over in Bakersfield. So been involved in farming for a long time, from citrus to some veg crops all the way into almonds, some pistachios and stuff like that. So just here to get a little bit more info on ComCat and kind of the deal with everything going on. So just glad to be here.
Okay, great. Okay, maybe I can start. I'm Thomas. I come from Germany. I'm the main developer of ComCat. I made my PhD on ComCat. I did my PhD in South Africa. I worked there at the university for 10 years together with Prof. Sieve. He's also my doctor father, and we developed ComCat to a product that it can be used. And I'm in this project now since 28 years. So I'm right from the start developing this project, and we are serving ComCat around the world from Chile up to north to the US. And we have now a very good client. I had this week a meeting in Costa Rica and all these countries. UPL, I guess you most know, UPL is one of the biggest fertilizer producer, and they are very interested to sell our products in the Caribbean, which I prefer to go then as a support. And yeah, we are in South Africa with our research. There we have more products, and you will hear more about other products from Rian. We have Engro, Cibazil, we have different types, adjuvants, silicon. But ComCat is one of our products. We are in Asia, quite big, China, Vietnam, Turkey, and in Europe. So we are quite all over the world, up to New Zealand with the product. Yeah, and maybe I will give you a short introduction to you. Then can you share my screen?
Yeah, you can share it.
Okay. 1 second.
Okay.
Okay, can you see it now?
Yes.
Okay. So I will tell you today a bit more about ComCat, how ComCat is working. Now it's the end. I have no idea why, but it happens. So I will give you a short introduction, then say something about the general mechanisms, application, stress, soil microorganisms, and the summary. And please stop me if you have any question in between. Everything is fine. I like it if we have a question, please come urgent with it and not wait up to the end. So the basics of ComCat started 1995. We as Agrarforum started 1998. And we have a lot of scientific research and proven effects worldwide. As I said earlier, we work quite along with Wilbur Ellis with the seed treatment for years. And on the bottom down, you see our factory here in Germany. It's extended now to triple of the size what you see in the pictures. In the new in the next year you will see different pictures, because now we want to make everything green outside and nice, and then we will enter the new pictures. On the right side you see our office and research center, and also production capacity in Bloemfontein, South Africa. This is right in the heart of South Africa, south of Johannesburg. And yes, I will tell you something about the general mechanisms. As I said earlier, crop health, improved quality, and higher yield. These are the key goals what a farmer wants. And ComCat is a bioextract, which is manufactured from special wild plants, which we grow here, which we extract in a special way. And yeah, it works as a catalyst. I don't know whether you know the term allelopathy. This means plant acts with each other, and we help to the plants to act in a way that they can defend themselves towards stress situations by activation the primary metabolic systems, photosynthesis and respiration. I will tell you later what we are doing in this case. And one of this is stress, drought stress, heat stress, water stress, salt stress, all these type of stress. We try to help the plants to fight a bit better against this. And most of the crops, and oversee many years, we saw that you get a higher yield as a benefit for a farmer, only to have better reaction on stress situation do not help. The farmer needs more yield, higher yield. And there's no added chemicals in ComCat. We only use special plant, and we extract the secondary plant compounds, and which are known for this to promote healthy growth, root growth, plant growth in total, from seedling up to the full-grown plant. And as I said earlier, it's from wild plants. The plants are not anymore wild. We cultivate them. We identified the plants. We selected the plants and we breed them now, but we breed them without any fertilizer, without any pesticides, without any watering. So, and the plant live in their normal area where they are grown, because we know that there are the best ingredients or the best molecules they produce where they naturally come from. And wild plants are known for their systemic acquired resistance. This means they have their own ability to resist against abiotic, which we looking for, and biotic stress. And it's a natural resistance mechanism. It's nothing unusual. Every plant has it. Every plant is able to protect itself up to a certain level. And most of the agricultural crops lost this ability because they are breeded for high yields. And this, on the other hand, costs some other energy. So the plant have no energy anymore to protect itself good, because they are breeded to give high yields. And with ComCat, we try to give some of these molecules back, or we activate some genes that they produce a molecule to protect itself. And this is part of our research, where we look for morphological root growth, root stimulation, seedling growth, and induction of flowering. We look for physiological ways, photosynthesis. If you have a higher one, you can get more sugars. Respiration, OPP pathway, more energy. I looked in my PhD on respiration, where we found that we increased in a seed of a pea 100% more energy. We got it. So it's fantastic. And we also look to sugar translocation, for example, in carrots, where the sugar is transported. We mark some glucose and apply together with ComCat, and we saw that it grows down to the roots. And whereas in the most of this untreated, the most of the sugar was still in the leaf, and then the ComCat was already in the carrots. And for sure, one of the part is the stress tolerance toward different stresses. And the bonus is improved yield and quality. How you can apply it? You can do a seed treatment direct. You can blend it with any normal treatment what you have. It can be treated just before planting at the applicator or coating. It can be as a foliar application on different crops, row crops, vegetable, fruits, or seedlings before you transplant it. And quite important is you can mix it with herbicides, fungicides, insecticides, and fertilizer. Herbicide, we do a lot of research in the past year that the combination of ComCat and herbicide have two effects. One effect is that the weeds are quicker killed, and the plants like soybean or rice or corn are more stable and are not so much affected by the herbicide. And it's known that a herbicide also affect the donor plant which we want to grow. And here in the detoxification, ComCat helps the plants. It can be applied via irrigation, pivot or drip irrigation. And what we are doing in special areas, we make a fertilizer coating, but it's a very limited market. We do it here in sports field. We are in several soccer stadiums in the Bundesliga. in Bremen, Hamburg, Leipzig, Gladbach, Wolfsburg. So we're in a quite range of soccer stadiums with our product, especially with a coated fertilizer. It's organic certified, OMRI, and according to a European standard, and also one ComCat version is CDFA certified since last year, if I remember correct, since last year. The mode of action, as I said earlier, we have secondary metabolites, which we get from plants which grow naturally and which are not supported by any chemical. So these are the natural protection of a plant. And when we apply it to seeds, leaves, we have a stabilizing effect on the cell membranes that we, for example, forces the plant to produce more vitamin E to protect the cells, especially under stress condition. And we have catalytic molecules. There the name comes from communication and catalyzation, that we forces the plants to produce some molecules which can force the plant growth. And why healthy plants? They adapt to their environment, and if you have a good healthy plants, the plants can absorb carbon dioxide, use sunlight and produce carbohydrates, protein, lipids, and water and nutrients from the soil through roots, have plant function and growth. And if the seedling starts slower, then they are more susceptible to pest or environmental stress. So here, the work of ComCat starts. If everything is 100%, well watered, optimum temperature, optimum fertilization, you don't need ComCat. But when you have it, and in the past years, more and more stress situation comes, but it doesn't mean that if you have no water, then ComCat will do a job. No, up to a certain level, this product can work. No product will help you if you have no water. And if you use it in a seed treatment, you can get a strong, healthy seedling, especially if you have stress conditions. We did some work with Wilbur Ellis, where we looked to cold, wet, growing situation, and there we help the plant to come quicker and stronger out of the soil. And afterwards, the farmer is able to manipulate the plant by giving the right fertilizer, the right amount of pesticide or water. But a seedling is like a young baby. It can be attacked, it can be ill. So the quicker and stronger it grows, the better your potential of yield will be. Here, a few examples of a quicker growth on rice. This is from Wilbur Ellis, where we looked here to cult stress. So here you can see a better growth, root and also top growth, so the plant can take better the situation. Here we have corn. Also you see quicker growth out of the soil, so the risk is less. Wheat, the same. You see clearly line where we use it. Soybean, we looked also to the rhizobium, to the buds, and here it was a trial where we looked to different concentration, and you see clearly what happens if we treat the seeds with rhizobium and ComCat, and it's really enormous. But the function of the roots, I know you know all what the fixation, absorption, conduction, storage, these are the main functions of roots. So if you have a good root system, you help the plant to grow better. And here a few examples of vegetables: lettuce, cauliflower, tomato, cabbage. We produce it in big pots, and then we looked afterwards what happened to the root system. We washed out of the soil, and we got these wonderful pictures of different root growth. Here we have a very nice story of a farmer in South Africa. It was a semi-irrigated trial. The farmer was willing to support us and said, okay, you get half of a pivot. The total pivot is 40 hectare, and he gave us 20 hectare. And he irrigated in the beginning, then he stops irrigation, and then the serious drought come. And then he phoned the prof in South Africa with whom I did the trial and said, you damaged my wheat. And he asked why, well, what happened? Then he said, in the control, all the plants started to forming ears, and the ComCat, you can't see one ear. And then the prof said, oh, wonderful news. This shows that the plants are under stress, they start forming ears. And then we wait 6 hours, prayed that this will come true, what we hoped. And here you see the results. When we came to the farmer was very, very happy. On the left side, you see the root system, left the control, right the treated one. And we have a better root system. But if you look to the top of the roots in the ComCat, you see a lot of soil is around it compared to the control. And this means the roots are very active, giving the soap out to the microorganism, and this glues then the soil to the roots. So you have a very, very good system, and they are still producing. And on the right side, you can see then the results. The ears are more equal, and the plant a bit higher, but we are not looking forward a higher the plant. We are looking more for the ears. And in this case, the farmer got about 600 kilograms per hectare more, and he was very, very happy. And yes, he's one of our good clients now, but this is since this was 15 years ago. Here you can see root development on rice. So, and function on leaves. I don't think that I have to explain to it. Photosynthesis, transpiration. One part maybe floral induction. There's not a lot of people know that in the leaves, they're producing a hormone florigene. And this is very important. So you need a healthy leaf that this production can be translocated later then to the buds. So it's quite important, and for sure food storage. So the bigger the leaf area or the healthier it is, the more food can be translocated, can be stored. Plants, plant growth, leaf development. The same is in spring. If you spray, then very quick the growth starts. So you come and it started to grow. Here you can see earlier flowering, and this was baby marrows, and here we could see quite well a huge increase, and later on also in the number of fruits. This is from South Korea, where we did trials with Fuji apples and application at the pre-bloom stage. And we have a higher number of flowers per branch. And important is that the stigma receptive was increased. This means you get a higher fertilization rate compared to the control. More fruits per tree where possible. And this is why we want a flower induction. The more flowers you have, the more potential, the more seeds you can have, and the higher your yield potential is. Crop resistance is quite important, and already in 1999 it was said that you lose up to 25% per year due to abnormal temperature, drought stress. So it's very important that you put the plant in a good position to fight better against stress situation. And we are not willing, or we can't afford yield losses like this. And here very short how ComCat works. ComCat we apply as a signal molecule, as an activator. We forces the plant to open genes to produce some molecules to protect itself, for example, proteins, enzymes, which can be used later on if the plant be attacked. And this we help the plant in their systemic acquired resistance, in their natural system. We don't give anything special, we only help their own system. And here we look to a few factors: drought, heat, cold, or herbicide stress. And this is a way, as I explained, we give a signal, the plant takes the signal, and it comes into the cell. Here, messenger RNA synthesis starts, enzymes, proteins are produced, for example, drought stress, vitamin E, cytokinin, which you can measure, and you protect the plant. And we did a trial already in 2001, 2001, 2, and 3 years at Virginia Tech, where we looked to turf under drought stress situation, and on the pictures you can see better root development. And what we were very interested in, looked into the plants, into the enzyme activity. And here we look to vitamin E, alpha tocopherol. And you can see under drought condition, we have 140% more vitamin E in the leaf. So we can protect the plant much, much, much better as a free radical catcher. We proved this also on the gene level in 2004 at the University in South Africa. In the model, this is Arabidopsis thaliana. This is where the most of the genome was identified at this stage. But we found at our research, after 30 minutes already, this gene, ATPH01 gene, is opened. It was a novel gene at this stage. And this gene, we found later, is also open if a drought stress situation comes. So this is part of the system, and we believe that we spray ComCat, and the ComCat opens special genes like this gene, and then you can have a better protection. We proved this also in Switzerland at the University of Geneva with Professor Strasser. I know him. He did a very good research on the live plants. He can measure the chlorophyll fluorescence by a JIP test, and then he puts the plants under drought stress, and you don't have to take a leaf. You can measure it over a period of time, over 14 days, the same plant without killing it, and you look for a performance index. And here you can see the performance index is 1. This means this is before the stress situation starts. And then you look a few days later how it developed, and you can see with ComCat, under middle and high drought stress, the plant is able to fight against this, and the control, this is the blue, it loses this performance index, so the started to battle. And we followed this for 15 days, and here you can see the first days the index is 1, and then we start with the drought stress, and 2 days later already the control plant loses this index, and the ComCat won't fight it, and we are nearly the whole 15 days over the line of the performance index 1. So we put them over a period of time in a better position to produce further. Here, sorghum, cold, wet situation. You can see on the right side, ComCat, the seeds are out of the soil, and on the left side, you can see already the roots, but no plant is out. So the plant are much stronger if they come out. And we did research on herbicides. We looked to different herbicide application with ComCat on maize here, for example, because we got the question, if we apply ComCat, do we make the weeds stronger? Also, yes, this is something what we said, okay, we will have a look to it. And in the first line, we could see in most of the cases, if we apply a herbicide alone, this is brown, for example, here we lose plant area, but if we add VC, this is ComCat, we increase 2,4-D, and in all the cases, we reduce the stress. We also looked, in this case, also to the general fungi and bacteria in the soil before treatment and after treatment. You see, we increase 8 weeks afterwards the total fungi and bacteria per gram soil. And methoxyline is a very, very difficult herbicide. If you have residues in your soil, your legumes can be killed or heavily affected. And we looked on soybean and groundnuts, and here you can see we have the young plant and the control. In the first season, we have nearly 2 grams, and in the control, it's nearly nothing. And if we see treatment and foliar application, we increased over 2 years. But for groundnuts, the foliar application nearly or is totally good for the plant, so no effect of residues in the soil. So methoxyline is a very tough thing. If you have residues in the soil, you can lose a lot of your yield, a lot of plants if you start in the next year these legumes. Then we looked also what happened to the to the weeds. We looked to one grass, and for 3 broadleaf, we looked to methoxyline as an active substance, glyphosate as an active substance. And here you can see what happens 14 days and 28 days after application. And you see with the herbicide, oh, go backwards, no, backwards. Here you can see that if you add ComCat to this herbicide, to the methoxyline in this case, for 2 or for most of this, we have a higher control rate compared to the treatment without ComCat. So this confirms our thinking that we help the plant in a better metabolism, and this means also wheat have a better metabolism. This means the herbicide is quicker in the plant and around the plant, so it can also die quicker. We confirmed this also with Panga Plus. So the same effect, if we add ComCat, we have a better control of the weeds. It's small, but important was that we don't make the weeds stronger, that you need more herbicide. No, it's other way around. And since a few years, we also look to the soil. What is with the soil health? What is with the microorganisms? And here, partners of us in Korea, which are specialized on bacillus growth, they did trials with Bacillus subtilis, and they looked to the growth. And here you can see 7 days after application of ComCat, what happens to the bacteria or the bacillus. We increased the number of Bacillus subtilis in the soil. It stays over 20 days, so we help the formation of the growth of Bacillus subtilis. And this was confirmed by one of our partners, Dr. Philippe, who is the producer of Ecoflora and Ecofauna. And he looked at different bacillus strains, and he looked the same that the rate of the turbidity of multiplication is increased. Here we have mycorrhiza fungi and ComCat and in the middle, and you can see clearly the advantage in the root growth. Here again is what happened to the rhizobium. Here we looked in South Africa to the rhizobium, but we repeat this trial in Brazil at the university, and they found the same, that we increased the nodules, these are the rhizobiums, significant compared to without inoculant, and with the inoculant alone. And also root, foliar, and leaf area, all a significant increase. So this confirmed our data, which we got in South Africa at our own research station. So coming quick to the end, ComCat is a stable formulated plant extracts from non-toxic wild plants, which can enhance growth. It's natural organic product. It contains only natural compounds, active substance, which we extracted from these plants. It's absorbed in the system and it helps the plant maintaining structure, function, especially stress conditions. We increase the metabolic processes, induce the induction of defense mechanisms. We have a positive effect on the microbial life. Applied with herbicide tended to reduce the negative effect of the herbicide on the grain crops, and it can improve development, produce higher yield and better quality in crops. And we always say ComCat is working when you are sleeping. Thank you for your attention. Now you can start with questions.
Alrighty. Does anyone? Yeah, go ahead.
What is the range of use rates given crops and methods of application? So I assume seed treatment would be minimal. In ground furrow or irrigation, drip irrigation, it'd be a different rate versus foliar application. What is the range of rates that you need to use to see the desired effects?
Lijuan, will you give the South African? I have the hectare, but you have the acre numbers.
The dosage, I talking about
Yes.
dosage.
Yes.
Okay, dosage for a foliar spray here is 20 grams for most of crops per acre, and then with drip irrigation would be 40 grams per acre.
30?
No, 40 for drip
40,
irrigation, okay. 20 for foliar.
Okay. What about pivot or flood irrigation?
Yeah, if pivot, it depends on the amount of water. If you're talking about
does it just matter on the nozzle?
No, it depends more on the water, the amount of water.
Gallons a minute. Okay.
Yes, but for example, if you use in South Africa 50,000 liter of water, there we would recommend 60 grams per acre.
Okay.
If you use this amount of water.
Okay.
So the higher volume you're using, you're going to want to increase your percentage of ComCat so it doesn't get too diluted. Okay.
Diluted?
Yes.
Yeah.
Yeah.
So like the guy spraying in West Texas at 10 gallons an acre, bare minimum.
If we're lucky, if we get 10 gallons a minute.
Well, 3 of it evaporates before it hits the leaves, so you're down to 7.
Right. Gotta love West Texas.
And the wind never blows, so it always goes
Yeah.
direct to the target too.
Right to it. Gets through the canopy and just goes right to the ground. Yep.
I think, well, Will, you also have a question about the adjuvant for ComCat as well?
Yeah, Ben told me the other day that you, I guess, I know Wilbur Ellis and Helena with Roundup. You know, we recommend pretty strong adjuvants with it, like brimstone, hellfire. They really heat up the Roundup, and I didn't know if there was a chance of degrading the activity of the ComCat or not.
Yeah, yes, Thomas. Will was asking.
Yes,
Do
sorry,
I
can
need to
you
repeat?
repeat? I just had a phone call from a guy who is in Boston, German, and
That's
sorry.
I know from my experience with Roundup applications like Wilbur Ellis and Helena, you know, we recommend a real strong adjuvant like brimstone, hellfire. Those both are they really heat up the mix, so to speak, to really um, cut through the waxy cuticle, the weeds, and try to keep the metals from tying up in the hard water and things like that. So I wondered if how stable is the ComCat going to be with something that, you know, that can be harsh for other things in the mix. What things do we need to worry about adding to the tank that may or may not decrease the activity of the ComCat?
This
Is there
is a very
anything
good
that we should
question.
worry about?
Yes, this is a very good question. So far, we don't see any negative effect. We tested it with a lot of different herbicides, fungicides in combination, and
Copper, because in the specialty crops down here, copper is very prevalently used to help with sterilizing. So, and I
yes.
know you got to be careful adding to the copper. Would the copper,
Now, we use copper here in apple, in organic apple farms. This is the only way how they can protect. And they combine ComCat with their copper, and we saw no negative
no
effect so
problem.
far.
Okay, all
No,
right, that's
no.
one thing I
Also,
wanted to
the
ask.
pH range can be alkaline to acid. So if you go in the pH between 5 and 8, we see no problems.
5 and 8, okay.
But you also can send me message later, WhatsApp, or you can send me an email. When we'll give you the email and make questions. If you don't have now a question, you can send me. I will try to follow up with the questions immediately if it comes. This is my job. I know it's sometimes difficult if you hear something, then come with questions. But I'm always able to answer also later. So if you don't have now a question, but in half an hour comes a question, send the message, and we will support.
What trials have you done on side by side or comparing competitor products to ComCat? Because I know Wilbur has some products that they would say claim to do similar things. So, or like Helena does too. Have you done any trials that kind of compare?
Yeah,
one has.
In the US, we didn't do a lot. We did some trials with Helena, and I think they showed very good results. And I think Lijuan will support with this data. And I think we performed very well compared to this. We did a lot of trials in South Africa where we used Calpac as a standard product, because you don't have a lot of products which have this biological ones. and also Messenger, and we are quite good in the results compared to these products.
Yeah, so we do have some of the trial results and also even like a field trials, with the farmers, compare, the ones without ComCat, the one use ComCat. We do have some of the trials results we can share with you.
Yeah, like Dr. Thomas, I was telling when I interviewed last week, I didn't want them to think I was peppering them with negative questions, but I know what growers think and how they're going to respond, and so I'm just trying to get out in front and try to get answers to the questions that I know I would face in the field here. So that's why
Yes.
I was wanting to know about
No. I like it. I like
but
it. This is something what I really prefer, because we also still can learn a lot. We don't know everything, so we can learn a lot. And especially if we come from the field, the questions are mostly the best questions.